Science and Technology Foundation. I'm really very happy to see you all here. So thank you. And with this, I will ask Mr. Suleiman to recite few ayah of Quran. Thank you.
وعنت الوجوه للحي القيوم وقد خاب من حمل ظلما ومن يعمل من الصالحات وهو مؤمن فلا يخاف ظلما وكذلك قرآنا عربيا وصرفنا فيه من الوعيد لعلهم يتقون أو يحدث لهم ذكرا فتعالى الله الحق ولا تعجل بالقرآن قبل أن يقضى إليك وحيه وقل رب زدني علما.
Thank you, Suleiman. Now we start the formal start or inauguration of the conference will be about 10 o'clock. So this is just starting the program. So I would invite Dr. DrLaythMula-Hussain as the chair of next session. Thank you.
Assalamu alaikum wa rahmatullah. Bismillah ar-Rahman ar-Rahim. Ladies and gentlemen, we are going to start the first session. May I invite Mr. Professor John Armstrong to the stage. Professor Armstrong is a consultant radiation oncologist and currently serves as a chairman of the Irish Cancer Society. He did his training at the Memorial Sloan Kettering Cancer Center, and he worked in Ireland after that for many years. He is widely recognized for his leadership in oncology and education and cancer advocacy. His work focuses on advancing high-quality, patient-centered cancer care, strengthening cancer policy, and promoting innovation and collaboration in oncology at national and international levels. Please join me to welcoming Mr. Armstrong.
Salam alaikum. Ladies and gentlemen, it's a great privilege and pleasure to be here today at this very auspicious and important meeting. My topic is embracing the future of radiation oncology. I want to begin with the story of a woman called Margaret Cleaves in New York. Over 100 years ago, around about the time Marie Curie was in New York, she was given a gift of radium, which she used in Columbia University for women with advanced cervix cancer. And she inserted this radium directly into the cervix of women who had potentially unresectable, incurable cancer. And she basically came up with a cure for locally advanced cervix cancer and published these successful results. However, she concluded at the end of her presentation that this technology could never be adopted because it's too expensive. And this concept that radiation therapy and the technology we use to deliver it is too expensive remains to this day the original sin of our specialty. So the question becomes, is it too expensive? And this graph shows you on the top the sales of cancer drugs from 2025 estimated going forward, global sales. And these sales are $192 billion per year, and they're going to rise and rise and rise. And in contrast, the sales of radiotherapy in 2025 are estimated to be 8.5 billion. So this is how it looks today. This is medical oncology, and this is radiation oncology. We are tiny, we are being dwarfed. And this imbalance creates enormous problems for society and for cancer care. It means that the drug companies, when they produce a new drug, attract the attention of all the media because they pay for it. Governments think the way forward is drugs. Why? Because drug companies build big factories. They provide employment. They pay tax. And when it comes to evidence, the drug companies are the only ones that can really afford to run the huge trials that prove that their next drug is going to work better than their last one. So this is a self-perpetuating problem. And in contrast, radiation oncology sales do not permit that kind of sponsorship. How many of you have ever been paid by Electa or Varian to run a clinical trial? I haven't. Every medical oncologist I know gets offered week in, week out participation in clinical trials by drug companies. So what are the effects of this radiotherapy underutilization? Because that's what we're dealing with. So in this slide, the optimal use or calculation of use is based upon the proven indications for radiotherapy correlated with the population and the types of cancer in that population. And in Australia, in 2015, that optimal estimate was 48%. But as you can see, only 25% of people are actually getting any radiation. And there are parameters that predict this. Being a man, being old, the type of disease, and living far away from a radiotherapy centre. Now, this is based on New South Wales, which is here. This is the populated part of Australia. This is a big empty space. So if you live up here, it's going to be even worse. And there are many parts of the world where populations are spread out and you can easily transport a drug to that location, but you cannot easily bring a patient for 8 weeks of radiotherapy to your location. So what are the impacts of this underutilization of radiation therapy? What does it do? Well, unnecessary surgery. So for instance, if a person can't come for 8 weeks of radiotherapy or 6 weeks of radiotherapy, maybe that man will have a prostatectomy and suffer all the consequences. If they have surgery and they don't get adjuvant radiotherapy afterwards, they may relapse, which is psychologically devastating and physically devastating if they require salvage therapy, and can, of course, lead to death. Because many of the reasons, the situations in which radiation is not being used are situations in which radiation improves overall survival added to any other available treatment. And of course, it also means that patients receive inadequate palliation. And it perpetuates the same problem we've already identified. Those patients who relapse are going to have second, third, fourth, and fifth line drugs with more and more drug expenditure and more cost to society. Now, I remember a time when we dreamt of having a drug such as Herceptin. We dreamt of finding a single drug that could target something other than the estrogen receptor or the androgen receptor, and we didn't have them. And along came Herceptin, and it seemed to be a golden era. The problem is that the cancers keep mutating and changing and changing and changing, and we chase a progressively smaller subgroup of patients with a unique molecular pathway, and that becomes more and more expensive. To find a pathway and drug it approach. And this is a picture of the genetic chromosomal abnormalities in a single prostate metastasis. Now, many of these changes are not going to provide the cancer with an advantage, a survival prospect, but many of these changes potentially will allow the cancer to self-fertilize and will then have to become a target for yet another and another and another and another drug. And we're trapped in this business model, in this biological business model, which is going to corrupt healthcare. So we need to take another view of it, and we need another vision for what we can do. So this is a slide detailing the story of a single man with a prostate cancer, treated with prostatectomy and ultimately dying. And at the beginning, he had a focus of Gleason 6. And what they did was they tracked the genetic abnormalities in all the different pieces of prostate cancer they got access to over the 17 years. He was treated with surgery, various drugs, palliative radiation, and eventually chemotherapy. And at this point, he had a biopsy in his lung. And that showed that this cancer bore the molecular genetic signature of the original P1, not the more aggressive histologies in the prostate, not the original lymph node metastatic disease. And when he died, these cancers that killed him at autopsy were clearly shown to have originated from that lung metastasis. So in theory, had you stopped that process at the point of the lung metastasis, he may never have died. This is a patient of mine I treated many years ago, a 65-year-old man who had an adenocarcinoma of the lung with a right upper lobe primary, mediastinal nodes, and a left adrenal metastasis. He was treated with cisplatin, Alimta, had a partial response. After 18 months, the mediastinal nodes were gone, but the primary tumor and the left adrenal are still there. So we gave him stereo, 60/3 to his lung, and we gave 40/5 to the left adrenal, and very importantly, dose painted off the stomach and off the spleen. He's alive 7 years later with no intervening therapy ever since. So it would appear as if we stopped his cancer in its tracks. This is the paper from Gomez about oligometastatic lung cancer, where patients were treated with chemotherapy to begin with. And if they responded, they were randomized to continue more chemotherapy or to have radiation to all their sites of disease, sometimes a bit of surgery thrown in. And this showed an overall survival advantage and a disease-free survival advantage. But what I want to draw your attention to, the new lesion-free survival, in other words, the occurrence of new metastases following this treatment. And these are the patients who continue on the drugs, okay? Drugs which were working, had induced a response, and yet did not stop new metastases from happening. And radiation, which is not a drug, which is not a systemic therapy, by killing the bulk of disease that was present, reduced the risk of developing new metastatic disease. So how is this happening? Is it simply mechanical? Samuel Hellman wrote a paper many years ago about breast cancer and came up with the term stopping it in its tracks. Are we just stopping the process in its tracks? Or are we generating some immune effects? The ARIOLE trial for prostate cancer looked at men with a limited volume of oligometastatic cancer who did not want chemical castration. They refused chemical castration and they didn't have a systemic option. So they were randomized 2-to-1 to either just be observed or to have stereo to their various oligometastatic deposits. And what you see is that progression-free survival was significantly enhanced in the patients who had stereo. And it meant a delay in the time for chemical castration, which was very valuable to those men. But what I really want to point out is this graph looking at T-cell clonal abundance. This is the control arm, and these are the individual patients. Not a lot happening, not a big difference at all in their immune profile. Why? Because no one's doing anything. Why should it change? And here are the patients being treated with radiation. And with a few exceptions where their T-cell clonal abundance dropped, you see a significant rise in T-cell clonal abundance. And this, of course, is a soft evidence of the concept of radiation altering the genetic or antigenetic expression of the cancer, rendering it more susceptible to the immune system. And this is happening in the absence of any immune systemic therapy. These patients are not getting pembrolizumab or anything like that. So we know about elective nodal irradiation, that sometimes it's a good thing. We've always done it in head and neck cancer. We have evidence that patients with breast cancer with node-positive disease and patients with high-risk localized prostate cancer patients have a benefit, a disease-free recurrence benefit, when they get elective nodal irradiation. So in those patients, those men and those women, you're radiating them all. And in a certain percentage of them, you are radiating lymph nodes that have cancer cells in them. And that's beneficial. The problem is the other 70% of patients that you're giving elective nodal irradiation, you're murdering their immune system. You're killing their lymph nodes. You're killing their lymphocytes. And yet it's overall beneficial. Now, wouldn't it be brilliant if we could tell which patients are which? Who are the patients who have some disease in the lymph nodes? We need to be able to image micrometastatic deposits in lymph nodes. And this is an interesting paper I found about an experiment on mice with tongue cancer, where they use an antibody against EGFR, made it radioactive, or sorry, fluorescent, and then dissected out the patient, the poor mouse, dissected out the poor mouse and looked at the presence of lymph node metastases correlated with the diagnostic image. And here you see a little micrometastasis or in a lymph node. Now, that's the prospect we need to reach for. That's where diagnostic imaging is a huge part of our future. We need to be able to image lymph nodes in human beings with very small deposits of metastatic disease, and then there will be no such thing as elective nodal irradiation. It will either be no nodal irradiation or it will be therapeutic nodal irradiation. And this brings me to the broader issue of what we're doing on a daily basis when we radiate each and every patient in your clinic. You don't know, and I don't know, whether you're having a good or a bad effect on their immune system and their immune system's ability to fight the cancer. And that may be absolutely core to the success or failure of radiation oncology. And this is a global question and has to be answered on a global basis by cooperation between organizations such as AMSTRO, ESTRO, and ASTRO. But as a specialty, worldwide, we need to do something to organize ourselves at the level of molecular research to understand really important questions like this. It's a global issue and it calls for a global coordinated response. I'm going to switch a little bit to exciting new technology, some of which exists and some of which exists somewhere up here in my head. So I'm going to start with protons. This is a recently published trial from the US, proton versus IMRT for oropharynx cancer. Stage 3 or 4, good numbers, standard chemotherapy radiation, and the 5-year overall survival for protons, 91% versus 81%, a striking 5-year overall survival advantage. IMRT, in other words, old-fashioned IMRT, was more toxic. It caused more serious events. Now, I put it to you that if a drug had caused a 10% survival advantage in head and neck cancer, every one of you would know about this. And every health authority in the world would know about it. And yet, how many of us in the audience today actually knew this result? And I think this highlights how even when we make great strides forward, we don't do anything about it. Who here has a proton machine ready to use? I don't. And that's the problem. We don't have the technology. Our technology is very hard to deliver, and I'll come back to that. I want to speculate a little bit going forward about the future. So imagine a prostate patient with systemic disease who is treated with lutetium. Trigger happy. Patient with metastatic disease who is treated with lutetium, and after the lutetium has a post-therapy PET. Imagine a linear accelerator living on that same gantry as the PET, and it's reading all the gamma signals coming back from the isolated micrometastatic, small macrometastatic deposits. And it's designed so that there and then, if that signal exceeds a certain threshold, it sends a boost, a stereotactic boost right back to where it came from. So A, you're putting the lutetium in there and radiating it internally, and B, you're zapping it with a highly focused beam, a micro beam. Now that could be a photon, it might be a proton. And you could even, in theory, get to the point of having single protons heading towards single cells. Now, I know that's almost impossible because I've read stuff about Rutherford, and Seifel Hook is having conniption fits when he hears me say this. But something like this can be done in the future. So this brings me to the concept of using radiation as a drug sensitizer. We all grew up knowing about drugs used as radiosensitizers. But here's the biological rationale for using things the other way around. We want to eliminate chemo-resistant clones before they proliferate, before they mutate. Local disease, whether it be the primary or the metastasis, can kill a patient. Get rid of it. Simple debulking may influence survival. But there's another model, another way that radiation may be used. And this can be driven by the financial consideration. There can be a drug, an expensive drug, which if you keep the patient on it for 4 years, they will probably live forever. But 4 years at what cost? And perhaps what we need to do is after 2 to 3 cycles of that drug therapy, we do a PET. And based upon the degree of response, we stop the drug and zap all their cancer with stereo, with VMAT, and with gamma-guided backfire, the term I've coined. Now, in that scenario, if we have radiation-sensitive disease, we may be using it on the basis of cost, duration, and reduction of toxicity. And if it's chemo-resistant, it may be because of efficacy. But what about if it's radio-resistant? Well, I'll put it to you that there is no single cell on planet Earth that can't be killed if the dose is high enough. And that's a challenge to the technology. And I think that technology will come. So what is the future we should embrace? And again, I want to reiterate, this isn't a question for AMSTRO, nor for ESTRO, nor for ASTRO. It's for a global collaboration. We need to improve advocacy for radiation on a national level in each individual country, on a regional level, and on an international level. And that message needs to be loud and clear and consistent and relentless at every chance we get. So governments know if you invest in radiation oncology, you get better value than drugs. We cure more patients than they do. And you will get a better return on your investment if you do that. Our manufacturers need to change their business model. Don't sell me a Linac. I want you to sell me an implementation package, a training package. I want a finished product. I want to be like a medical oncologist who gets Herceptin and gets told, okay, put the needle in the vein, now inject. There you go. That's Herceptin. I want it to be that simple for us to overcome the challenges of adopting transformative technologies. On a separate vein, we need a major investment in radiation oncology laboratory research for the fundamental mechanisms of how radiation works and for how it impacts the immune system. And we need to think of and develop trials which test the concept of radiation as a drug sensitizer. And we need to work harder to integrate radioisotope therapy into the other forms of radiation we now have. That cannot be a nuclear medicine specialty. That needs to be our specialty. If we do these things, we will deliver a better future for our patients. And we will avoid the prospect of crocodilumab devouring gamma dinner. And finally, over the decades, I have heard many colleagues say that the technology has reached its limits. There's nothing else to be got from technology. It's time to look in another direction. I put it to you that the only true limitation is that person's inability to imagine a better future for our patients. Thank you very much. Shukran.